In this webinar, we're going to talk about proteomes and the resources in UniProt. My name is Pedro Raposo, and I'm a senior curator and bioinformatician. We also have Tanusha, which is an analyse fellow, and Swati, which is a senior software engineer. So the outline of this talk is going to be, we're going to start with the different UniProt resources, and then focus more specifically on the proteomes and how we can explore these so that you can take the most of the information that you can have from this resource to do your own analysis. And we'll have a bit of a demo at the end. So the UniProt is a consortium that is formed from 3 different teams. We have EMBL EBI from the UK and Cambridge, and then we have also SIB, which is the Swiss Informatics Institute of Bioinformatics, and PIR, which is the Protein Information Resource in America. And these 3 teams work together to provide all the services that you can find in our website. The UniProt is a website that has multiple resources, and they are interconnected within themselves. So UniProt starts by gathering information from external sources. Some of these external sources is NSCC, which is a collaboration between DDBJ, GenBank, and ENA. So all of these share the same genomic data, but also we gather information from more specific databases such as RefSeq and Assemble. And once we get the genomic information from these databases alongside with their translated proteins, then we are going to archive those within UniPark, which is our sequence archive. This is a non-redundant sequence archive, which means that for each sequence of amino acids, for each protein sequence, then we have multiple entries coming from different organisms. And once we have those protein sequences, we can create accessions in the UniProt Knowledgebase. The UniProt Knowledgebase is our most used resource, since it contains most of the information, such as protein annotation, function, and comments coming from expert curators from UniProt. They have the responsibility of reviewing scientific papers in order to provide you as much data as possible to these proteins. The UniProt Knowledgebase is divided into 2 data sets, SwissProt and Tremble. SwissProt are the proteins that we just mentioned. These are the ones that they are manually reviewed by curators. And then we have Tremble, which are entries in which the annotation is created through automatic annotations. Then we can organize the UniProtKB accessions in UniRef by clustering them together according to their sequence identity. But also we can group them by proteomes, gathering them by the genome assembly itself. So a proteome at the end will contain all proteins coming from that genome assembly. You can think of a proteome similar to a genome. In the same way, you have a set of genes that will create a genome assembly. You can also think as a proteome as all the translatable proteins from that genome assembly. Among these proteomes, we have a special set called the reference proteomes. So at the moment, we have more than 1 million different proteomes in the UniProt website, but we have only 37,000 reference proteomes, which correspond more or less to 34,000 species. And you can think of a reference proteome similar to a reference genome. These are the proteomes that contain the best quality in protein annotation, and they represent the landmark for that taxonomic group. So you can think of as the most or the recommended set of proteins for that species in UniProt Knowledgebase. One thing that is worthwhile noting is that the UniProt knowledge base only contains proteins coming from reference proteomes. This was a transition that we've been going through in the last year. Since previously the UniProt knowledge base would contain proteins from all proteomes, but now we transition into a UniProtKB exclusively with proteins from references. Also, we have other proteins that are biologically interesting, even though they might not be part of these reference proteomes, such as, for example, SwissProt. All the SwissProt are going to be maintained in the knowledge base, regardless if they come from a reference proteome or not. This transition was well documented through our blog post, through our release notes and forthcoming changes. But also it was documented in a webinar that I added a link here. And we have released a manuscript that is under review at the moment, where we describe the methodology for the selection of reference proteomes. So the transition was possible due to a creation of the workflow that selects the reference proteomes. So this is a simple schema where you can see that the UniProt knowledge base before contained proteins from reference proteomes and also from non-reference proteomes. And you can see this by the color code, where the translucent organisms here represent the non-reference proteomes, and you can see that for example, for some species, we would contain multiple references and multiple non-references. And we idealize that the UniProt knowledge base would be better suited if we only have at least one reference for each species. And also we would not contain multiple references for the same species as well, so that we could direct you into the best data that we could provide you. And so at the end, the UniProt knowledge base has one reference for all species, and ideally we will have just one references, but for some species, because these can be very diverse biologically when it comes to the proteins, then we can have multiple references. This is our land page on the website. This is what you see if you search UniProt, and right from the get-go, there's a couple of things that I would like to point out. The first is that you have the search bar in the middle of the page, in which you can search a keyword, a specific protein, or an organism, and you will be redirected into a search result page where you can see that keyword being found in those entries. Then at the very top, you can see multiple tools that you can use, such as BLAST and Align, that will help your protein analysis. But also you can see at the bottom of the page that you have the four main resources from UniProt, and this is UniProt Knowledgebase, the Proteomes, UniRef, and UniPark. But in this presentation, we are going to focus on the Proteomes resource. So if you click on it, then you will be redirected into the Proteomes land page. And here you can have a brief introduction to what is a proteome, what does it mean to UniProt, and how can you search them. You have some simple statistics, such as what is the number of reference proteomes and the number of non-reference proteomes, but also the taxonomic distribution among them. And also some simple documentation for you to get started with if you have any questions. The help pages are going to be explored at the end of this presentation, but these ones are the ones that we recommend for you to start with if you have any questions. This is a page of an example proteome. In this case, it's the human proteome. And you can see that at the very top, we have the protein ID, proteome ID, which it's unique for each proteome. you'll have additional information such as the status. You can see that this specific proteome is a reference, but also you have different information that you can find it useful when exploring it, such as the number of entries or the number of UniProt Knowledgebase entries, the taxonomy, where this comes from, the genome assembly, where these proteins are translated from, but also some metrics on completeness, such as the Busco score. For some proteomes, you have a small description, and you will also have how is the proteome formed. So what are the components in this case? What are the chromosomes within it? We are going to go through an example in the demo at the end of the presentation. If we explore the proteomes resource on our website, you will find that we have more than 1 million. And in this result page, where you can find all of them, you have here the human proteome, in which we just had a look at. So each of these lines represent a different one, and you have some basic information on this table where it's customizable. You can add other information such as the genome assembly, or you can remove some of the ones that you can see here. And this help in this search is supported by filtering through different criteria. One of them would be the taxonomy. So you can find all the proteins belonging to bacteria or viruses or archaea, or even for specific species. Also, you can filter by the status of the proteome. So this is the categories in which the proteome is inserted into. And we will have a look into what are these categories. The first one, as the main one, the one that we mentioned, is reference proteome. So again, these are the proteomes that have the best quality and represent best the protein space of that species. Then you have the non-reference proteomes. These are the ones that were not selected as references. The sequences are still archived in UniProt. As you remember, in the UniProt Knowledgebase, we will only have proteins coming from references. So all the other proteins will still have proteins, not in the UniProt Knowledgebase, but in UniProt, our archive resource. Excluded proteomes are also archived in UniProt, and these are proteomes that aren't fit to be presented in the UniProt Knowledgebase. Most of them fail quality control checks. So let's get back into our human proteome as an example of the reference proteome. As mentioned before, you can see the status on the top left side, where you can see this is a reference. And also you can see that it has 147,506 entries in the knowledge base. And if you click on this number, then you will have access to all of those proteins. And as mentioned before, these proteins can be inserted into two categories, Swisspot and Tremble. And you can see at the very top that 20,416 are Swisspot, which means that these have been manually reviewed by curators in UniProt. And also we have a large quantity from Tremble. On non-reference proteomes, you have the same status here. And if you click on the number of entries, now you can see that they are associated with UniProt. Instead of you being redirected into the knowledge base, you are going to be redirected into UniProt as well. And the same thing happens for excluded proteomes, where these proteins are also going to be redirected into UniProt once you click them. So this is an example where you can search all the sequences in UniProt. You can see that we have 2,816 here. And if you click on one of those, you can have access to to a page where it has annotations coming from Unifier, which is our automatic annotation system that provides annotation to UniProt. On the top, you can see that the status states that this sequence was removed from the knowledge base because it does belong to an excluded proteome. So in this page, you can find plenty of information on this protein. Of course, it doesn't have the same amount of scrutiny as if it would be in the knowledge base, but UniProt is also getting improved due to this big transition that we had on the proteomes and the knowledge base. So we are providing more and more annotation on these pages. They are still very reliable, and you can get plenty of information from UniProt. Other information could be, for example, family on domains. This is a page that I would encourage you to explore, but it's a bit out of the scope on this presentation. I just want to note that whenever you see the keyword automatic annotation associated with annotation in UniProt, it means that this was generated using Unifier. So Unifier is the software that was developed by UniProt, which contains the two main automatic annotation systems developed by UniProt, which are ARBA and UniRule. And this aspect was already covered in another webinar that we have at the beginning of this year for automatic annotation systems. Also, we have help pages if you are interested in learning a bit more about this. There are a couple of things that I would like to show you before we start the demo. The first one is that we now have this metric called proteome similarity, in which we compare proteomes to the reference proteomes of the same species. So in this case, these proteome ID UP00002285, it's a non-reference proteome. So you might think as, okay, this is a non-reference proteome, so it does not belong to the UniProt knowledge base. But if you are still interested in this proteome, because you are studying this genome assembly, for example, then you would be interested to see, which are the proteomes that are present in the knowledge base similar to this one. So at the bottom of the page, you can see that we do have two reference proteins for the species, where you can see this in the right side of the slide, and you can see the similarity of the protein that we are now seeing, the one that ends with 285, to the other ones. So you can see that it's 88% similar to the first reference here mentioned, and 84.8% similar to the second one. So these will help you to determine which are the proteomes that are best suited for you when you don't have this one available in the knowledge base. Which ones should you start looking at? We also have similarity not only from non-reference proteomes to reference proteomes, but between reference proteomes as well. So in this example, we are having a look into the first reference proteome of this species, and we can see that there is a similarity percentage to the other reference. The other thing I would like to note is we also developed, we revamped pan proteomes. So you can see here in this example on E. coli that on the top right side of the page, you can see that it says that this proteome is part of an E. coli pan proteome. So if you click on it, you'll have some files that you can download when it comes to proteomes. But before that, I'd just like to mention that pan proteome is simply the reflection of the proteome diversity of that same species. So in the same sense that you'll have access to all proteins from the reference in the knowledge base, the pan proteomes will allow you to have a bit more in-depth proteome comparison of one species in the pan-proteome. So these would allow you to compare different proteins of the same species and determine which are the proteins, for example, that are rare or common between them, but also studying individually the non-reference proteins as well. So the pan-proteomes were revamped using MM62 clustering, which is a software developed by Martin Staniger Labs. And the outputs that we offer at the moment are three. So we have the FASTA files in which for each protein cluster, we select the most representative one, and we add to these species pan-proteome FASTA file, and it has some information. One of them, which might be important to you, is the protein population frequency, in which tells you if this protein is rare or is quite common within the species. Then we have a matrix in which tells if a gene is present or absent within each proteome, and some general statistics on the pan-proteome itself. All of those can be downloaded using our FTP, so we would encourage you for you to have a look at if you are curious. And if you have any feedback, not only on pan-proteins, but also on the reference, you can also contact us. If you have any other questions outside of this webinar, not only when it comes to proteomes, but any other topic on UniProt, you can go to the help pages. We do have a help resource in which you can search for a keyword, and you'll find all the pages associated with it. And all of them are pretty much interconnected. So even if you start something as generic as the proteomes page, then you will find the associated pages such as what is a reference proteome, how was it selected, for example, and then one of the characteristics is Buzzcode score, so you can see what is the quality process and what is the methodology that we used, for example. So all of these questions can be answered through our help pages. This was one of them that we updated. So all the help pages that are associated with proteomes, pan-proteomes, were all updated due to this change that we've been transitioning into. So these are up-to-date pages that will help you understand what the current state of the resources. As mentioned before, if you are interested in how the reference proteome was selected, this was work that we have been developing for quite some time. We have a technical webinar on it that was recorded, so you can access it. But also we have a preprint on these novel methods. And if you have any additional questions that could not be answered either by this webinar or the help pages, you can always reach out to us. Not only that, but if you have any feedback or you see something that doesn't seem right in our website, you can always contact us. We are pretty quick responding. So we encourage you for you to reach out to us. And now I'm going to go through a quick demo. So this is our page where you can see what I described before. You have here the search bar. You have the 4 different resources, but also you have different resources that I did not mention before, which is taxonomy. It is gathered from an NCBI taxonomy, for example, or specific keywords that we use. or, for example, subcellular locations of different proteins and the interaction between them, and automatic annotation, the UniRule and Arbo that I was talking about. Also, you have different news that we are constantly releasing. For every different release, we provide a release note, but also forthcoming changes in the next one. We have here some of the tools that are at the very top of the page as well to help you analyze your protein studies. And at the very bottom, you have a summary of those, and also you have the help here, or at the very top as well. So let's dive into the Proteome resource. So if you click on it, you'll head up to the line page. And from there on, you can simply go to the search and search, for example, for, let's search for human. You have here all the results associated with Homo sapiens. You can easily identify the one that is a reference proteome by this red icon with an R. That means that is a reference. You click on it, and this is the proteome page. I'll go through a bit more specifically what are all of this information. So as mentioned before, this is the ID, this is a status, this is the number of proteins that are in the knowledge base because this is a reference. And if you click on it, you'll be redirected into the knowledge base where you can have access to all the proteins. And if you are familiar with UniProt, you know that if you click on an accession, you have all the information associated with it. So the function comments, you'll have some gene ontology terms that were added by it through manual or automatic processes. You have some more information on the protein names and recommended alternative names, short names as well, gene names, the taxonomy, and much more information. You also have subcellular location. You have different cross-references to all different databases, including the ones from where this protein came from. In this case, it comes from Ensemble. So this is a bit. Oh, you also have 3D structure of the protein, either from PDB or from AlphaFold. So this is something that it is definitely explored in other webinars and courses that we have in UniProt, but this is just to show you that you can have access to those from the proteome. The objective is of the proteome is to gather all these proteins coming from the same genome assembly. Then you have the gene count, and these correspond to the canonical proteins of this proteome, the taxonomy and the taxonomy ID. You'll have access to the genome assembly if you click on it. So you can see that it comes from Ensemble in this case. And if you click on it, you have access to where these proteins originally came from. Remember that we gather information from INACC. So it's mainly ENA, DDBJ, and GenBank, but also we get information from RefSeq and Ensemble. And so these databases provide not only the genomic coordinates, but also the translated proteins. If you want to get a bit more information on the genome itself, I would recommend to go to these resources. Then you have the genome representation if it's full or partial, and then two metrics for completeness. So you have the CPD and Bosco score, which allows you to infer the completeness of a proteome. Then, as mentioned before, for some proteins you have a description, the components. You can see the different chromosomes, but also some additional genomic information such as mitochondrion. You'll have the publications associated with it. And at the end, you have the similarity score, but this is not possible for human because human is the only reference for that species. Let's go to another one that I have over here. This one is an excluded proteome, and this is because we have it as a low-quality proteome. So excluded are proteins that are not suited to be in the knowledge base. And if you want to gather a bit more information on it, you can click here and you'll see the different reasoning why we don't have this in, why we have this as excluded. So this example, you can go down and it says proteome failed UniProt quality and control analysis. So this one failed the analysis, which means that it was not deemed good enough to have in the knowledge base. You can see that Bosco score is quite low as expected. And if you click on the number of entries, it will go to UniPark instead of UniProt Knowledgebase. And here you can see all the sequences associated with it. So if you click in one of them, you can see that we can have multiple cross-references to different sources. So as mentioned before, the UniPark is a non-redundant database, which means that for each protein sequence, we will have multiple cross-references coming from different proteomes or even from different sources. In this case, you can see this exact same protein coming from a different proteome here. So if you click on it, you can see that it is the reference proteome for that particular species. Remember that all of these can come from different species, but in this case, they come from the same one, it's just different strains. If you click on the second one, this is the one coming from the proteome that has been excluded. So if you go here, it's the proteome that we were looking at. And here you can see that this protein comes from, belongs to an excluded proteome. And these are the annotations associated with it. In this case, these were imported. Then you have some more information on UniPark. Not only it's an archive database, but also contains plenty of information as well if you would like to have a look at. And this is the example that I was showing in the slides. If you click on it, you can see that this was generated by automatic annotation. In this case, it was specifically provided by an arbor rule, which is an automatic system that we have. Then you have the sequence here and potentially some similar proteins from UniRef, which is our clustering resource. One last thing I would like to show you. So you can access the Proteomes resource either by going to our website and clicking here, or you can go here and search for Proteomes. And in this case, I'm going to search for E. coli. So you can see here that we have multiple reference proteomes associated with these keywords. But remember that I just searched for the keywords Escherichia coli. So it means that you can see here, for example, some phages, because they are associated with the bacteria. Remember that you can filter by the origin or the superkingdom of the proteome. In this case, I want the bacteria. And here now you can see that we have 9 reference proteomes. If you click on one of them, if you go to the very bottom of the page, you can see that these current reference proteins that we are having a look at, we have the similarity to the other 8 reference proteomes. And they are ordered, which means that this first one is the one that is most similar to the one that we are currently seeing. There is one last thing that I would like to show you, which is, let's go back. Another way of searching reference proteins that sometimes we get some emails from people that get a bit confused by our search results is that sometimes you see more proteins, more reference proteins, sometimes you see less and people get a bit confused. So remember that I'm just searching for the keywords. If I want to search by the species, I can go, instead of clicking the proteome ID, I can click on the species, but I clicked on a specific strain, K12, you can see here. So if I want to go a level up from the strain to the species, I can go here to the lineage and click on E. coli. Now, the pages that I'm showing you currently, this is coming from the taxonomy resource that we have. And from here, you can search the proteomes associated with it. So you can click here. But now some people come to this page and ask themselves, why don't we have the references here? I know that there are 9 E. coli reference proteomes, and this is because we are just searching on the species level, not below. So if you want to search below, you need to click here. It says expand search on the tax ID 562, which is E. coli, to include lower taxonomy ranks. So if you click on it, now you're searching not only on the species, but below, so all the strains. And here you'll find the correct number, 9 reference proteomes. And if you click in one of those, another thing that I want to show you before I go is that the genome assembly from this proteome does not come from ensemble like human, but comes from ENA or INSTC. So if you click on it, you will be redirected into the page where the genome came from. You can explore the chromosomes. In this case, it's just one, but also other quality metrics such as N50 or the number of contigs, for example. You can explore on the genome level where what is the quality of this genome. And with that, I will finish the demo presentation. And I want to thank the funding that we are having throughout the years, including INH, and also the different teams supporting all the work that we can provide to you by our website. We have more EMBL EBI webinars. As I mentioned before, we have already webinars on specifically on the technicalities on how we selected the reference proteomes. But also we have other related to UniProt, such as automatic annotation, or even a webinar specifically designed to show you how you can search different proteins and how you can maybe use BLAST and Align and different tools that we have in our website. And these have been recorded for this year from 2025-2026. And you can access all of those webinars here, but also we will start having the new series in October of this year. And also we have EMBL EBI also has webinars for other different teams and groups, not only UniProt. And with this, I finish my presentation. And if anyone has some questions, we will go through them.
Petra, thank you so much. It was a great talk, and thank you so much for the demo as well. We can start with the first question: how creation of proteins that are in UniProt database works?
Every time we start by importing information from external databases, as mentioned, we create these UniPark entries, and these are going to be created as Tremble entries in the UniProtKB knowledge base. So these Tremble entries won't have initially any manual information. Then we have different teams of expert curators, not only in EMBL EBI, but also from in SIB in Switzerland, that these curators that are experts on different fields and different taxa will go through the available literature in order to infer what is the function and the biological context of this protein. And they will add as much information as possible to these entries. So once that information is added to the Tremble, then these entries will be created or will be transformed into SwissProt entries.
Thank you so much. We have another question asking, how can we select most common SNPs for a specific protein for a study?
SNPs.
SNPs.
I'm definitely not the right person to talk about SNPs. We do have people that are.
You can show the variants tab. At least they know that.
Yeah,
That's very
yeah, that's a good idea. Let's go for one in human. Always has. Okay, variant viewer. Yeah, so in these, sorry, you are able to see all the different SNPs that have been reported to this protein, and you can see the pathogenicity of them here, and also some more information below.
Another question about if a person has their own proteome from their experiments, can they compare that to a reference proteome in UniProt?
That would not be possible at the moment. What I'm thinking is that you can download all the proteomes and associated proteins from our website for that species, and you can do your own comparison using AAI, for example, average amino acid identity, or you can even cluster the proteins like we do using MM6, for example. That would be a fair comparison. If you manage to update your genome, your genome assembly plus all the translated proteins into NSCC, then we will import it to UniProt. But you need to wait a few releases for that to happen.
Yeah, I just add that we encourage users to go and look at UniPark because it's being actively developed. So a lot of the information that was removed from the knowledge base is not lost. It very much still stays in UniPark, and we are continuously and actively developing it. So you have the structure viewer, you have the feature viewer, so it should look like a UniProt style knowledge base page. So we would encourage users to go and explore that rather than panicking that my protein is not there in UniProt.